This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 888, recorded on April 12th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's 62 Fahrenheit at the moment, mostly cloudy. We had some rain earlier, but we're in that spring phase of, sometimes cold and rainy, sometimes threatening to be nice weather.
Can you imagine if it had been August 8th today?
Yes, that would have been perfect.
888. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. I have the other type of spring weather in that it is 70 Fahrenheit, 21 Celsius, and beautifully sunny outside.
I don't know what it is. When I came this morning, it was raining, and now I haven't looked outdoors, so I have no idea if it's nice. Yeah, the weather says it's sunny. We have a guest today who is returning for a second time. He is from Atlanta, Georgia. Richard Plemper, welcome back.
Thank you. I'm very glad to be here.
So I last recorded with you in October 2019 at Georgia State University. And I didn't release...
There have been no interesting developments in virology since then.
Yeah.
So we thought we'd have you back on to catch up.
I didn't release that episode until the following July, because in January we started doing all COVID. And one of the things we did is a paper of yours from, 2021. Yeah. Showing that Molnupiravir blocks transmission in ferrets. And I said, well, we'll have to get Richard back. And so here we are, Richard. Welcome back.
Yes, thank you. And really, and when you were in Atlanta, we talked actually about the drug, which was then not called Molnupiravir, at a study that we had just done against influenza virus. And so we demonstrated in the ferret model that what is now called Molnupiravir is actually very orally efficacious against influenza. And at the time, that was interesting, but nobody knew how important antivirals would become, right?
Yeah, that's amazing.
2 months down the line.
Who knew?
When you were thinking about the development of the drug now known as Molnupiravir, what viruses were you originally trying to combat? Was it originally supposed to be an influenza antiviral, or which viruses were you trying to hit then?
So the plan was actually to launch the initial clinical trial with, we called it at the time, EIDD 2801. So that is a joint development product with my long-term colleagues at the Emory Institute for Drug Development. And so EIDD 2801 or Molnupiravir, the original plan was to run a clinical trial against influenza virus. And that was actually scheduled for the second half of 2020, a phase 1 safety trial. And obviously that then got very rapidly superseded by the coronavirus development. But so originally it really was influenza. So 2801 is very broad spectrum activity, and we are coming from the negative strand respiratory virus field when looking at that. But we knew already before the pandemic that the drug is not only active against negative strand RNA viruses, but again, for clinical trials or clinical indication when developing drugs, it's very important that one actually has a clinical trial, or that a drug can be tested in a meaningful trial. And that when looking for more at some of the extreme or of pandemic potential pathogens in before a pandemic hits or in interpandemic years, it's very difficult to design a clinical trial. Mm-hmm. And so that drove our interest to influenza.
What year was the drug developed at the Emory Institute?
So we started working on 2801 is a prodrug. as an ester form that makes it orally bioavailable in higher mammals, so in NHPs and as we now know in humans. The original drug was something we called N4-hydroxycytidine, or in our nomenclature EIDD 1931, and that we had the first paper on 1931, I believe, in 2015. That was also for flu and for RSV, for respiratory syncytial virus. And so that looked only at rodent models, so at mouse models, where 1931 is actually quite decently orally bioavailable. But as I said, not in higher mammals. So from that, really the work started in about 2012, 2013. And the ester form, so the orally available product 2801 Molnupiravir, that we first had in the lab maybe, I think, early 2018.
So the original screens were done. So you decided to look for polymerase inhibitors, obviously, because you're making a nucleoside analog, right. and the original screens were done with influenza virus, right?
Right. Was influenza and RSV.
RSV. Okay. When you did those original screens, did you design a drug that you thought would react with the polymerase as a nucleoside inhibitor, or were you just taking sort of compound libraries and looking for inhibitors? Yeah,
so in the lab, we actually pursue both approaches. But as Vincent just said, so 1931 is a nucleoside analog. And really, if you think about chemical space and drug libraries, so for nucleoside analogs, the chemical space is much narrower than for non-nucleoside small drug-like molecules, where libraries can easily go into the hundreds of thousands or millions of compounds. That is typically not happening with nucleoside analogs. So much smaller libraries, is really no high throughput was necessary. We do screen high throughput in my lab, but for nucleoside analog libraries, we essentially look at something of a few hundred compounds.
Right, because in this case, you know basically the structure that you're looking for is going to look like a nucleoside, and so then you build a library of compounds around that.
Right. And the meaningful chemical space, so the number of analogs of variants of the chemotypes of your nucleosides, right, that you can create is really quite narrow. So it's not tens of thousands of different combinations that would be meaningful to test.
And then this, just for folks who haven't kept up necessarily with the drug development process, this prodrug strategy is a well-established thing in the drug industry, where you've got something, it seems to work, but it's not easy to deliver orally. And so then you add to it and modify it and make it into something that when it's digested, it goes into the bloodstream in the actual form that you need, right?
That's exactly right. And so during GI absorption, the ester is hydrolyzed. And so what we detected then in PK, so in pharmacokinetic studies in plasma, is also of NHPs, is only the hydrolyzed original form, so 1931. And that, of course, is a nucleobase, right? So it's N4-hydroxycytidine is a nucleobase, the actual active compound that the viral polymerase would incorporate. That's the triphosphate form. And so that the anabolism to the triphosphate happens by host kinases in the actual target cell in which the virus,
This is one reason why works well against the virus in cells is not really necessarily a slam dunk, right? That you've got this whole road to follow.
Yeah, that is really the challenge. So with nucleoside analogs, not only, of course, does it need to be recognized by the viral polymerase as an acceptable substrate, and ideally with preference by the viral polymerase and not by host polymerases, but also the compound needs to be efficiently anabolized. So meaning the cellular machinery also needs to recognize it as an acceptable nucleobase and convert it to the triphosphate. And so that, from the discovery aspect, that can often be challenging, because if there is no activity, then the question is it simply because the polymerase doesn't accept it, or the polymerase would love it, but it is not converted to the triphosphate? And that can so to differentiate between the two alternatives, and then to ideally through MedChem to try and address whatever the problem may be, that can be very challenging. And then on top of that, as you said, for us it is imperative that direct acting antivirals are orally available. So that was one of our major developmental objectives, simply because we think that in particular for these rapidly replicating RNA viruses, where the therapeutic window, so the time available for efficacious treatment, is typically very narrow, that we really want to and need to reach out patients for an efficient therapeutic approach, and that we feel is can best be achieved with an orally available drug. And definitely, as we've seen with remdesivir, for example, that really, I think, is the central liability of remdesivir, that IV delivery is just not on a population basis, is just not feasible to outpatients.
Right.
Yeah. Yeah. So Molnupiravir is incorporated as a C and then mistemplates, right? That's the mechanism of action?
Yeah, so it can, after incorporation, it can spontaneously tautomerize, so it can change its appearance, its structure, and as a consequence of that, it base pairs alternatively either properly as a C or it base pairs as a U. And so that results then in at the next amplification step of the viral genome, that results in mutagenesis, random mutagenesis.
And so at what point in the pandemic you'd already been working with the drug? When did you decide to start looking at SARS-CoV-2?
Actually, coronaviruses. So Mark Dennison had already looked at coronaviruses in cell culture, and that was in 2019. It was kind of more as an effort to close the loop. We have a broad spectrum antiviral, and what's really what are what's the true indication range?
How broad is your spectrum? Yeah,
exactly, and defining the limits of that. And so Mark had a very nice study at the end of 2019 that essentially shows that in cell culture he has activity against beta coronas, and also which in particular, because of the proofreading activity of the coronas, right, cannot be taken for granted, that a nucleoside analog would actually be efficacious and not recognized as not natural and be excised. And so that was a very nice study that essentially established basic proof of concept in cell culture. What we then really did, and coming from the influenza ferret model, was to apply our experience with ferrets to SARS-CoV-2, and so establish our transmission model in the lab, and then demonstrate against, at the time, the original Washington 1 isolate, that the drug is actually efficacious in vivo against the virus.
And so obviously you have a BSL-3 there that you did these experiments in, right?
Yes. So actually we have high-end max containment. And so we were very nicely set up for the work, and the institution really has really risen, I think, quite admirably to the challenge. So we have greatly expanded, obviously have redirected all high containment work to pandemic response work, but also in particular moving ferrets into ABSL3. Ferrets are large animals, and that just brings some challenges under high containment conditions. And so I think it really helped a lot that we have worked for years with ferrets, that there was lots of experience under ABSL2 with the influenza work, but still it takes it to the next level. And it really required quite an effort from our biosafety officers and the animal resource team, that have made that possible. So they were highly supportive.
So you mentioned some of the previous research looking at Molnupiravir and other coronaviruses. How broad is this drug against coronaviruses, and could it be something that could be useful in the future?
So far
already
we have
thinking of the next pandemic. Yes,
right. I mean, who would have thought, right? Who would have picked in 2019 beta coronavirus as anyway, right? I mean, maybe some people, but I think if you would do a poll, probably not the majority. But so we have so far for the coronas, we of course follow through with activity against different variants, and have tested that in different animal models now in ferrets, and we also recently in a study that at the moment is still only a preprint, but we look quite a bit in dwarf hamsters. So these are the tiny hamsters. They are about the size of mice, or a bit smaller. And they come down with severe lung disease. So with acute lung injury, and challenge depends a bit on the variant, but is lethal within, if untreated, typically within 3 to 6 days. So it's essentially an endpoint model for severe COVID, and the lungs are truly, so there is massive, major macroscopic tissue injury, and of course, in the histopath.
And is that, do you know if that's primarily caused by virus or immune response?
So it's exceptionally acute, right? If you go into the most aggressive virus we found is actually with the Delta VOC in this model. And so we have first lethality within 2 to 2.5 days after intranasal inoculation. So massive cellular infiltration, massive viral pneumonia. So clearly we have a major immune response, but also very high viral load in the lungs.
Okay.
So it's not like the inflammatory disease in humans, which takes longer, right?
Right. So this was, and what we wanted to set up was a model that really looks at an endpoint at severe acute respiratory failure, and what are the limitations for treatment with a direct acting antiviral, right? Not in immune modulation treatment, but with a direct acting drug. Where do we reach the limits of the therapeutic window where we can actually change outcome? But so coming back, sorry, I totally missed or dropped your question, right? So we have tested in those models. We have tested all of the current VOCs, and so far we have not found a beta corona that would be less sensitive, and that again through work of colleagues like Ralph Barek's lab and Mark have looked at MERS and at SARS-1, I believe, and also have seen consistent activity, and that I think is very encouraging in terms of future activity of Molnupiravir against variants that may emerge down the line.
Right.
Have you tried to select resistant variants in the lab?
Yeah, we did. Actually, we and others. So, of course, we tried this, again, coming from the influenza field. We tried this extensively with influenza. We tried this extensively with RSV. And others have tried for other targets. I think Mark, in his original paper, already mentioned he tried for beta coronas. But and so far, really, nobody has identified definitely not robust escape. With nucleoside analogs, we see typically at best, if you look at other drugs, we see moderate or intermediate resistance. But with Molnupiravir, so far, it beats the rule that an antiviral that does not induce resistance is not an antiviral. So far, we have not been able to identify a resistant mutant. And that I'm pretty confident, because we do resistance profiling is something that we do routinely, and usually we do this quite successfully. So I don't think that is entirely due to our incompetence of creating escape mutations. So I think the genetic barrier against resistance is really unusually high.
So you think that any resistant variant is simply not fit enough to propagate?
Yeah, I think that so the real problem, I believe, at the time of incorporation, so before it tautomerizes, it's chemically really very close to cytidine. And I think that for the polymerase to be able, I mean, it's no problem to counter-select against the drug, right? But to counter-select and still be able to recognize endogenous cytidine or CTP and incorporate that seems to be a real chemical challenge.
Which the cell can do, otherwise you'd have horrible side effects, but the cellular equipment is much more elaborate.
So there you come to an interesting question, right? That is, so what's the selectivity of the drug? And we can in vitro assays, in vitro polymerase assays, right? Purified polymerase components and synthetic, so artificial RNA templates. And so if we compare their selectivity over the endogenous, so over CTP, then there is, we can see a preference of the viral polymerase, but is for the drug compared to host, but is not massive. Meaning the selectivity does not come from host RNA polymerases somehow magically not recognizing the drug. I really think the main difference is that we, meaning mammalian cells, do not amplify based on RNA templates.
Right.
While the virus, as an RNA virus, they're in the line of business of obviously doing that. And I think, so the catastrophic effect for the virus, that really kicks in when amplifying after incorporation. Right? That is when the mutations emerge. For the host, we would, if it's incorporated into, let's say, into mRNAs, and it tautomerizes, can we produce garbage mRNAs at very high drug concentrations? We can demonstrate this. That happens, right? And so at some point, then you also run into measurable cytotoxic concentrations. But that is of course, way above the efficacious concentration, the antiviral concentration. And I think it's also, typically mRNAs are very short-lived.
Yeah.
And so, I mean...
And to what extent if it's a messenger RNA, its job is to be translated by a ribosome. Right. Is the ribosome going to be affected by this change? Because it's based on base pairing. We think it does. So we think that if we force it into mRNA, so again through extremely high concentrations, then we see that actually protein biosynthesis is compromised. And okay.
That suggests that.
That's such a good point. I hadn't really thought of it that way. That's sort of making me rethink a whole lot of things here. So I'm really glad that you told us about that. I know there are also some papers that have sort of tried to say that molnupiravir also can lead to changes in DNA. Yes. What are your thoughts about that? Is there anything to that?
Really, the best data set for that are the data that Merck has released, and they actually have released an enormous amount of data from their safety studies, also the preclinical safety studies that they have conducted in 2020 and 2021. And so the answer really is, I think is we like with every medication, we clearly look at the risk-benefit ratio here, and it has to be some risk assessment. And I think that is, I mean, that applies to almost everything in life, right? But definitely to any form of pharmaceutical intervention, right? Every time we take a therapeutic. So the acute, from now a lot of patient experience, and also so molnupiravir is actually taken quite a bit. What I hear in Europe. And so I have family in Europe. And so some of them actually have taken the drug after a positive test and were, because some additional risk factors. And so this was for me anecdotal evidence, but very direct. So very, very unusual that we see actually this really, right? We cannot ask the ferrets. We look very critically, but how they really feel, right? So that feedback we typically don't get. And so what we see in terms of acute or immediate side effects, that really seems to be negligible, if any. And that matches the all animal data in multiple species that we have. So it's very well tolerated at the efficacious dose. The real question is now, what are the long-term risks? And it is true that to some very low degree, as we know, also ribonucleotides can and are incorporated into DNA, right? It's like, and so could this result in a catastrophic event? Now, the data Merck has released very strongly argue against that, but I think that is just there is some residual risk where really one needs to make an assessment, right? What is the acute risk of this patient to progress to severe and potentially life-threatening disease versus what is a long-term consequence of the drug?
Right.
What is long-term? How many years are we talking about, do you think?
Yeah, I don't know. Look, how often, probably in the past few years, much less, but how often have you boarded a plane for an intercontinental flight? And what
Sure.
that is a risk assessment, right?
Yeah, of course.
The benefit is obviously you like to go where you're going. And the risk is, of course, you get some measurable radiation dose, right?
Mm-hmm.
How will that ever affect you? Probably not, right? But maybe, and in what years, I think is impossible to answer.
Well, and when was the last time you ate some bacon?
Yes.
I mean, so if we're talking about mutagens, life is mutagenic.
Yes.
And then the question is this drug adding to your total mutagenesis load to a point that you need to worry about it versus the risk of progression to severe COVID-19? And obviously, the data so far and the FDA assessment is based on the voluminous amounts of data Merck produced on this was that this was a reasonable trade-off. So it's unlike some of the discussion about potential risks of some things for interventions. This is rationally based, right? There is mutagenic potential, but it seems worth that risk in the situation.
So if you look at it again from a data perspective, then the safety data are really all what I have seen that I'm privy to has been done very solid. So there were no corners cut. And I think again Merck was very open. So if you have followed the public hearing end of last year, that was an unusual, I think, an unusual transparency from the company's side in really making the data sets available. And I think those data were very encouraging, and in particular when really considering that for people with underlying complications, conditions that put them at risk for advance to severe COVID, that really the benefit can be major.
Right.
Isn't ribavirin a mutagen also, and it's been used extensively in Hep C before direct acting antivirals, right? There was no known issue with that one, right?
Yeah, ribavirin is this ugly drug, right? multiple, polypharmacological, right? But so does lots of things. But it's also discussed as mutagen, and the same is T705, right? Favipiravir acts as a mutagen and is originally for influenza conditionally approved in Japan as for severe pandemic influenza.
But not in the US, right? It was never approved.
No, but also in terms of efficacy, I was never terribly convinced by favipiravir. So we have used it as some of our control as a control in some of our experiments.
Yeah.
And one needs to dose extremely high. So at some point, the question is how realistic is that as a human therapeutic? But it has been given also in desperate situations, right? It has been dosed at exceptionally high concentrations, and yeah. So I however. That may be with efficacy or not, but definitely MOA. So the mechanism of action is very, very similar, is based on a mutagenic effect. Yeah.
So was favipiravir tested against SARS-CoV-2?
It was. I'm sure. I mean, you named the drug.
Everything was tested against SARS-CoV-2.
I more at some point wondered how often do we really want to retest, like we collectively, right, as the scientific community, the same library of just a few thousand approved drugs against the same viral target and hope for a different outcome. I mean, at some point, that becomes an interesting exercise, right? But so favipiravir definitely was tested, but as far as I don't quite recall whether it was either not active or required exceptionally high concentration.
Okay.
Yeah, but so obviously it really there was no benefit.
So when is molnupiravir given? How long after what symptom onset is it have to be given?
Yeah, so I think coming from the influenza field, right? SARS is the much easier target, right? Because with flu, typically you go to bed healthy, you wake up dead, right? This is like extremely rapid onset. While with SARS, we actually have several days typically where a patient from infection to clinical signs to a week later advance to severe disease. I think ideal is, and now with the new technologies that the pandemic really has brought, right? So our testing capacity, testing infrastructure that just didn't exist before, plus contact tracing approaches that also really were developed only during the pandemic. I think that now we should have, in the ideal situation, one can identify a positive or get a positive test result even before the onset of clinical signs. And for a patient that belongs to an elevated risk group, I think the best would be to start treatment right at that stage. Because for a direct acting antiviral, right, before the virus load ramps up, that really is when the impact is greatest, and when very likely any form of productive virus replication in that host can be completely suppressed.
Well, and from your ferret work, it looks like in that case, getting it within 36 hours of infection was a really good idea.
Right, but in the ferrets, disease progresses faster.
Progresses faster, right? But it emphasizes the earlier the better.
So exactly, I think that's really the bottom line, right? For these acute viral infections, treat early and treat hard, and any approach of let's wait whether it really gets bad or not, then essentially, I mean, I think you alluded to that earlier, that really a lot of severe clinical signs that's driven by the immunopathogenesis, but that means that the viral load is either completely down or already in drastic decline. So then one really needs to ask, with a direct-acting antiviral, what do we want to treat, right? So if keeping virus load down, that has to happen early. I think at the late stages, then it's immune modulatory therapy.
Yeah, obviously, if someone is hospitalized, it's too late for an antiviral.
Right.
Most likely, right.
Which gets back to the chief problem of remdesivir, because people are not getting an injected drug until they're hospitalized.
Yeah, that's right.
Yes. Yeah, then it's time for immune modulators, right? To prevent.
Well, I think that was the problem with remdesivir, that most people were getting it in hospital and it wasn't working well. And then they tried some earlier pre-hospitalized treatments, which seemed to give a little bit of benefit, right? If I'm not mistaken.
That is absolutely correct. And also, I mean, Molnupiravir, the first clinical trial was actually to hospitalized patients, and that trial was not encouraging.
Yeah.
The data for looking at non-hospitalized patients, and the initial trial was up to 5 days after onset of clinical signs. Mm-hmm. So that was the Merck trial. Interestingly, Pfizer's Paxlovid trial was up to 3 days after onset of clinical signs for enrollment. And I think that actually really makes a difference, because if you look, I mean, viruses have a tendency to that they live in log scales, right? And so, as we know, and so 2 days on a log scale growth curve make a dramatic difference in virus load. And so it's obviously with clinical trials are very complex. And so to compare one to the other there's never all the parameters are identical. And so then the direct comparison, this is better, that is better, I think is very challenging, right? That's the beauty of the animal models, that we can really run a clean side by side.
Yeah.
And that would be a very interesting study if you really want to drill down. Is there a substantial, like a significant difference in efficacy? And I from the data I've seen so far, I'd be very surprised if there is.
So since the initial trials, Molnupiravir has an EUA at the moment, is that correct?
That's correct.
So have there been follow-ups looking at effectiveness in the real world? Do we know how it's doing, for example?
Yes, so again, there is definitely anecdotal evidence is coming, and I see. So what I have heard is that has been used quite a bit in India early on, mm-hmm. and those data were very encouraging, but it's very difficult because again, when humans are messy. Right?
Yes.
So there are lots, I mean, as we know. So it's again, I think it's very difficult to really get a clear picture at what is experience of, so what is based on very small anecdotal evidence or what is kind of a preconceived idea and what are really hard data. I think so far, I believe the clinical data are encouraging, in particular if treatment is started early. Mm-hmm. Right. And that I think is essentially, that's the same with obviously Paxlovid. Paxlovid has a different mechanism. But I think is still the same. It's a direct acting antiviral targeting protease, and so ultimately bringing virus load down or keeping it down if given early. And I think that again the data show that starting treatment early is the way to go. And
both Molnupiravir and Paxlovid are pills, so you can take them orally. Right.
Exactly. So that was the again for us the major objective, developmental objective, and that I think holds true for an influenza drug, and definitely holds true for a SARS-CoV-2 drug. That really one needs to be able to reach the outpatients efficiently.
Yeah.
And of course, a pill also has the advantages that is of more logistical advantages. That is shelf stable, storage is not an issue, distribution is not an issue. Yeah. That really I think is that are important factors, and for both for Molnupiravir and Paxlovid, the PK properties require that the drug is taken twice daily. Mm-hmm. Really, in terms of compliance, once daily would of course be preferable, but that so far, we cannot maintain drug levels long enough in the exposure levels in the tissues to control virus replication, and so meaning there is really no way around with these chemotypes of twice daily dosing. But we are working, and we just earlier this year we published a different nucleoside analog. That's our next generation drug that doesn't have a name yet. We call this EADD 2749 or 4-fluorouridine. And that drug we are again very excited about because it is also broadly active. It has a different mechanism of activity to Molnupiravir. We've shown it is actually a delayed chain terminator, so it's not mutagenic, but is active against coronaviruses, so against SARS-CoV-2, and and that's why I bring it up now. It is actually has a much longer half-life in the tissues. So it is suitable in all our animal studies. We dose it once daily, and we can show in our pharmacokinetic studies that we actually maintain full control of virus replication through this 24-hour cycle.
And that's orally bioavailable.
That is orally, yeah.
So this is related to Molnupiravir or distinct?
That's distinct. So that's a uridine analog. I mean, it's a nucleoside analog, right? So we are still in the nucleoside analog corner, if you want. But obviously, it's a very, very, very different, chemically very different, right? It's uridine based, and as I said, the mechanism is different. So this one is a delayed chain terminator.
It's like the remdesivir that causes a delayed clash with the.
That's exactly. So what we think is that after incorporation, and we in the study we from earlier this year we've shown. So again, this is tested in in vitro RDRP assays or RNA dependent RNA polymerase assays with and we did this in this paper with both an RSV polymerase and SARS-CoV-2 polymerase, and what we can show is that if you look at different templates, that in particular after tandem incorporations, we get preferred termination several nucleotides downstream. So incorporation of the analog, and then the polymerase incorporates with preference up to 3 follow downstream nucleotides and then stalls. And that's a classical delayed chain termination mechanism, and very likely reflects changes in RNA secondary structure by the incorporated analog. And so then the polymerase runs into a problem downstream because it cannot deal with these structural changes. And or through tandem incorporation, that doesn't need to be immediate repeat, but can be incorporated and then an endogenous nucleotide is incorporated, and then again an analog of the drug that amplifies the effect.
And this is active against SARS-CoV-2. Sorry, Alan.
Okay.
Yes, it is. So it is in ferrets. So we test again. We use the ferret model for SARS, and we use for RSV we use a mouse model, and in both cases highly efficacious.
And being uracil, would this reduce the chance of DNA mutagenesis?
Yeah, I mean,
if anybody's concerned about that.
If anybody is concerned about that, but yeah. So I would hope that the real alleviating factor here is that hopefully gives people some peace that the mechanism is really not mutagenesis. The mechanism is chain termination, right? So
sure.
We would hope that we don't even have to come near that
right.
So this brings up an interesting question. Is there space for additional SARS-CoV-2 antivirals, or do we have enough? What are your thoughts?
So, I mean, definitely more are in development. Gilead, we've worked, so my lab has worked a bit with Gilead, again, using the ferret model for an orally bioavailable variant of remdesivir, or rather a drug that so a prodrug form that is that ultimately in vivo in the body is metabolized to the same active form like remdesivir is. Okay, and so a variant of that is in clinical trials at the moment, and I'm pretty confident that will be pursued, and there are others in trials. I think clearly there is more space for and need for additional therapeutics. But I think that also one has to keep in mind that the next round of clinical trials will not be against placebo any longer. So that will be against standard of care that is now, I think we have established. And so it will be, meaning the bar is higher, right? When one needs to really show an improvement. And I think that may limit the ultimately the number of drugs that we'll see fully approved on the market.
So explain how that works. If you have a standard of care and then you want to try a new drug, you have to test it against the standard of care or together with?
Well, you cannot. Your reference group will not be placebo treated group any longer, right? So meaning you really need to establish improved efficacy of the new over the existing.
But you can have a group that just gets your new drug, right?
Right. Yeah. That you can. Yeah, of course. Yeah.
Okay.
I mean, depending on the indication. I mean, the FDA might not be keen on, we're only going to give the experimental drug to these people to, when there's a highly efficacious way to prevent their death otherwise. But in this case, the Molnupiravir arm or whatever, it's not as if that's a slam dunk, and it's not as if these people were doomed otherwise. It's probably you'd be able to set up a trial where your experimental drug is exclusive in the other arm.
There's room to look for superiority.
Yes.
But that is, you're right.
Well put.
Right. But very, very important point, right? That of course the design of the trial needs to meet ethical standards.
Yeah,
and absolutely. So if you have a patient group that is at risk of life-threatening disease, and you have standard of care established with which that could be mitigated, then one cannot see, let's see what happens with the unknown. That is very true.
This is the kind of thing you might want to do in, say, a young, healthy population that you would treat with Molnupiravir to slow spread and reduce symptoms, et cetera. And
Right.
Do you think there will be combination trials going forward of a nucleoside and a protease inhibitor, for example?
Yeah, I think that is very likely, and is at least from discussion that I hear, I think is definitely considered. Mm-hmm. Again, drug combinations are. There's of course also risk. I mean, we typically look or hope for synergy, either additive effects or synergistic effects. Synergistic is always that's of course what everybody wants to see, right? That's
a positive synergistic effect anyway.
I'm about that's why I bring it up. So what is less discussed is that there is also negative synergy, right?
Yes,
so we can also have an amplification of adverse events, of side effects, and so I think that really will be. So it will require new trials of the combination, obviously, right? So we had the monotherapy trials, the safety profiles, but it will require a safety profile also of the combination. So it's not, I think it's very likely, but it's not just a little achievement or
Sure.
low hanging fruit, right?
Right.
That we can say, okay, no, we have this is safe, that is safe, so we can throw them together, and can be sure nothing.
Pharmaceutical science is littered with combinations of drugs that are a really bad idea. Yes.
Yes.
I think it's important to point out, because I get questions from people. Okay, why don't we combine these now that we have two?
Right.
No, wait, you have to test them properly.
Yeah, I mean, you see, right? So coming back to the Paxlovid example, right? I mean, Paxlovid is actually a combination, right, of the protease inhibitor and of ritonavir. And what really I think is lost a bit in the discussion is that the ritonavir component, just as a little bit of background, is required to improve exposure of the actually active protein inhibitor, so to improve its PK properties. Otherwise, it would be metabolized too rapidly. And unfortunately, it does not only do that for the protease inhibitor, but it does that also for many other approved drugs, right? So it just interferes with drug metabolism. And that is really a contraindication of Paxlovid with many at risk of severe COVID as at-risk groups, right? So many, for example, cardio medications are contraindicated because suddenly drug levels would be when Paxlovid, so when ritonavir is taken, would be drastically changed.
Yeah.
So I think those effects also need to be considered very carefully, right? That we, when combining drugs, and in particular when changing metabolism, that even established safety profiles can completely change because suddenly half-life and exposure levels of the individual drugs are changed.
And those can go either way. There are many drug interactions where there's an inhibition of an enzyme that would normally break down some other drug, and now the patient has an astronomically high concentration of the drug that's toxic.
Right. Exactly.
And there are others where you inhibit an enzyme that's necessary for metabolism of the drug, and now they have a non-therapeutic dose of the other drug.
Right.
And it's also possible that you've got a nucleoside inhibitor. You think you know what its mechanism of action is, but that doesn't mean that it's the only thing in the entire body that it hits. It could be stepping on some enzyme that we haven't even heard of that's involved in ways we didn't even realize for some other drug. So we do have to tread carefully with that.
Vincent mentioned ribavirin, which is the prime example,
right?
Of it. Of a drug that in addition to its direct mechanism or the one of the best understood, let's say, has many other effects. So absolutely correct. Yeah, it comes down to the very simple conclusion. It is complex,
yes.
Right? And we really need to tread very carefully, as you said, because unexpected catastrophic events could happen.
I like the way you put it. It's a dirty drug. I wanted to ask you about the oral, your paper on the prodrug of remdesivir. We actually did this on TWiV a couple of weeks ago, and I had a question. I didn't know the answer to it. So now you're here. Let me ask you. So you have this oral version, which looks like it was around at the same time as remdesivir, but the decision was made to go with remdesivir. But it looks like it was much more active remdesivir than the oral version. Is that true, or what was the history there?
The oral version, so much more active in what model? So
in that paper, the remdesivir, the real remdesivir, seems to have a higher, the micromolar you need is much lower. I think it's about 10-fold lower. I think these were in cell culture experiments, basically, than the oral version. And I just wondered if that was the reason they went with a remdesivir, which was intravenous but more active.
So the oral versions are newer forms that did not exist at the original time of the remdesivir development.
Okay.
What we have seen, I mean, ultimately, so one has to, I believe one has to be very careful with cell culture experiments, in particular when we try to directly convert cell culture active concentrations, EC50s, EC90s, to predict efficacy in vivo. Just because the availability and the metabolism profile is are such major players in determining what tissue exposure can we actually really achieve, right? In cell culture, can essentially throw as much drug onto the cells as we feel or we can get into solution. In vivo, that's very different. And so based on the data that we've seen or the studies we've done, so we did a comparison with IV remdesivir, and the oral drug in vivo is more potent. But I think really what is ultimately the deciding factor again is that for most patients, I think IV is not an option before they are hospitalized.
Right.
Sure.
Right. And that I think is going to be game changing. Or again, that was what I think was game changing with Paxlovid, Molnupiravir, and for remdesivir, but this oral form is going to be game changing. That one can actually efficiently reach outpatients.
Okay. So the oral remdesivir is in humans now. You said it's in trial.
So a form of that, a slight, so a variant, but of the orally available remdesivir is in a phase 1 trial. Yeah.
But now these were, you were doing this work before the pandemic and focusing on flu, which, speaking for myself, really had my bet for next big pandemic. So, don't take my predictions for anything. But are you... it might be nice to have more drugs for SARS-CoV-2, but these broad spectrum drugs seem like a very generally useful thing to have in the pharmacopoeia. And are you getting back to that now and working on flu, measles, et cetera?
So this is really where 2749 came from. So this more recent analog, right? So my personal pet model here is that if we, in terms of, everybody talks about improving pandemic preparedness now, and I think that is a major objective, right? Or more trivially said, right? How can we prevent that we are caught as flat-footed again as we were 2 years ago? Right. And I think that it seems like that our ability to predict what will be the next pandemic threat is limited. Right. And so a broad spectrum, I see a major improvement from a drug development, antiviral drug development side for improving pandemic preparedness, that we have a panel of first-line defense drugs. And I think they need to be orally available. They need to be broad spectrum, or at least broaden spectrum. I think they should be ideally overlapping spectrums, but not identical. And coming back to the risk benefit, one just needs to accept, I think, that there is a certain risk. There is a risk with vaccination. There is a risk with therapeutics, but that the liabilities, so contraindications that we have, as we just discussed, for Paxlovid, or also potential risk for certain patient subgroups, they are also ideally are not overlapping. So that, I think, will give us the best arsenal to, as a first-line defense, to reach as many different patient subgroups with different needs, with different therapeutic needs in an event, a pandemic, or a new, let's say, a new potentially catastrophic zoonotic transmission event happens, right? Because that's really what stands at the beginning.
Right.
And what that means is also, for these drugs, we have to have an indication that can actually be used to seek approval for human use before the pandemic hits. Because that really is what we have learned, right? With both Paxlovid and Molnupiravir, we were really far advanced. And it just, so the compound existed, and it was very straightforward to get different animal model efficacy data. But even with this enormous pressure and unprecedented accelerated timelines to approval, it still took about 2 years, right? Or a good year and a half. And I think if we want to have a chance to maybe stamp out a mounting pandemic before it, or keep it local before we have global catastrophic proportions, then this panel of drug needs to be pre-approved.
So what kinds of indications could be used for that pre-approval?
Well, maybe flu?
There may be flu, right?
we have a flu pandemic every year, right? That's
right. So where we have established protocols for clinical trials, that I think is really very important. And yeah, influenza gives us that. For influenza, we also even have challenge models, right? I think if we talk about, so originally I'm a paramyxovirus guy, right? So for the paramyxo and high pathogenic, obviously Henipa virus, so the Henipas come to mind. And there we have outbreaks, and we have recurring outbreaks, very worrying, and obviously catastrophically high case fatality rates, right? But to organize a clinical trial against Nipah, that is very, very challenging. It would be much more feasible, for example, to have a broad spectrum drug to test it using established clinical trial mechanisms, pipelines against influenza, and then have it approved, have the safety profile, and consider it as an off-label candidate in case of one of these more pathogenic, more challenging, but less predictive viruses emerge.
And for coronaviruses, you could actually use the common cold coronas as a challenge study, couldn't you?
Yes.
Which nobody would have funded 3 years ago, of course, because why? But now I think you might be able to write that grant.
Yes, although now everybody has, we have so much invested into high containment labs, right? So,
right.
So it's, I don't know. OC43 is how much of, I don't know how much traction it would get versus the request, talking about what is fundable or what is an compelling application that people say, no, I really want to see this to be proof of concept against the real pathogen and not a surrogate. You can argue it either way, right?
So do you think if we had Molnupiravir or Paxlovid in December 2019, it could have stopped the virus getting out of China?
So I was in a discussion just a few days ago where we exactly tried to get a consensus on what could have been done, right? And so the answer is we couldn't get a consensus. So that's, but I think at least we would have means to really give it a serious try. Yeah. Right. I think what also needs to be then recognized is, I think the proportions of the challenge needs to be recognized, right? Because that goes directly into your risk-benefit assessment. So if you would, in retrospect, everything is clear, right? But in December 2019, let's say we would have had a drug like Molnupiravir with certain risk associated with it, as we discussed. Would the perceived threat at that time have been large enough that we say, okay, we need to go all in because that's our probably last chance to control this regionally? And who makes that decision then?
No,
right?
It would not. And I think we actually have the example of what happened in different countries to tell us that decision would not have been made in the U.S. or in Europe, for that matter, because we saw very early in the pandemic countries that had experienced SARS-1 in a significant way firsthand had a very, very different response from countries that hadn't had that experience. And so, South Korea, just absolutely locked everything down and stopped the virus. Because they had that prior experience, they had laid the groundwork for it, and they understood the risk. And I think worldwide, that was just not broadly understood that that was the game we were playing.
Right. But then really the question is the next pandemic challenge, right?
Yes.
And I think it's a matter
Now that we
of,
all do understand the stakes.
yeah, do we? I mean, really?
It depends on how long it is
Yes,
before
that's
that.
right. That's exactly right.
The time frame is going to matter a lot.
If it's 50 years, it'll be forgotten.
It'll be forgotten.
Yeah, but it's not 50 years. I mean, that I think we can be pretty sure, right? But I mean, again, a different discussion. And the question was, everything is coronavirus. So somebody asked me, do we care about influenza any longer? And I think some of us do,
yes.
right? So that about collective memory, right? We don't even need 50 years. I think collective memory is much shorter than that.
Yes.
That is worrying.
Well, 50, I say 50 because really the last pandemic that was as bad was 1918, right?
Right. Okay. Yeah.
So, but that's a different era. We're in the modern era, and so hopefully we remember.
Well, and that's the last respiratory pandemic. There is, that whole HIV thing.
Yes.
Yeah, there's that as well. Yeah, I agree. But I think these are really. I think you're all right. We would not have said this is going to be a big one in December and instead start using the drug. No, because so many countries didn't, as Alan said. But and Richard, you're right. In hindsight, sure, but that's not how it works.
Well, and for a drug that needs to be given in the active viral phase, you'd also need the testing capacity, which we empirically did not have.
Yeah.
Right.
That's a great point. Good points. All right, Richard, thank you so much for joining us today. Richard Plemper from Georgia State University. Appreciate you joining us, Richard.
Thank you very much. It was a great pleasure talking with you.
Thank you.
And
thank you. I learned so much.
This was very educational, yes.
Keep it up, Richard.
Do my best. Thank you.
I'm so glad that we had that discussion before I did my antivirals lecture in virology next week.
I just did mine on last Wednesday. Too late. But I have all those drugs in it.
That was a really cool perspective to have. That was great.
He's terrific. I remember him, and when I visited, we talked about flu and measles. Actually, has a paper on a measles antiviral, which is interesting, right? Because I want to know, why would you need that?
Why would you bother, right? You've got a vaccine that works.
But a lot of people don't get vaccine.
Yeah.
All right, so let's do a couple of emails here. Brianne, no, I want you to take which one? I want Brianne to take. There's one I want Brianne to take here. Which one is it? Which one is it?
I was guessing it was the first one.
Yeah, the first one. Go ahead. Yeah. Take the first one.
Mark writes, love your podcast. I'm not in the business, just a curious civilian who appreciates the breadth and depth you bring to various topics. I don't know if you take listener questions, but if you haven't already, I'd be curious to learn about, in quotes, immune fatigue as it relates to multiple COVID-19 vaccinations. I've vaccinated and boosted Johnson & Johnson and Pfizer. I see Pfizer is already putting the wheels in motion for authorization of a second booster and have followed Israel's policy of doing exactly that for folks that are older or have comorbidities. Even as the CDC noodles over it, the San Francisco Department of Public Health has authorized and recommended a second booster for anyone, regardless of age or comorbidities, who began their immunizations with J&J. No reasoning shared yet. Second mRNA booster to be 5 months after the first. I'm 56 and in that boat. I'm doing a full month of work travel beginning mid-April, and I think I'm going to get that second booster this weekend. My only concern has been the light chatter about possible immune fatigue from getting so many jabs. Is that genuinely a thing or just a postulate? Cheers and keep up the good work, Mark. So I am aware of something that I guess could be related to immune fatigue happening with very long-term chronic infections with an antigen or a virus that you have with you for years and years and years in your life. However, with a vaccine-induced antigen, I have not seen any data on any kind of immune fatigue or what I would say, if I'm trying to extrapolate to other things, exhaustion in the immune response. I think that if you think about this in your daily life, you are encountering some microbes pretty frequently throughout your life. And you don't tend to see exhaustion unless it is a long-term with you every day thing, like a chronic infection. With acute infections, I am not aware of any evidence of immune fatigue, and I would not worry about that with these vaccines.
And in terms of the sources for this immune fatigue theory, this is a sort of anti-vax adjacent argument. It comes up a lot in discussions like, oh, that's too many shots too soon. This is in too close succession. Natural immunity is better because it's somehow gentler. It ties in with that whole viewpoint. I don't know if that's necessarily where Mark got the information, but that is
right. It's always surprising to me to think about natural immunity being gentler, because it comes with the risk
of disease.
And when students ask me about, how many antigens at once is too many for getting a vaccine, because this is sort of where that comes in.
Yeah.
You could possibly get too many antigens. I usually then will move in the lecture room under an air vent, where air is getting blown into the room, and point out that I am probably getting as many, if not more, antigens while standing under that air vent than I might be if getting vaccinated with a few vaccines.
When I pick up one of the cats to put them in the case and they scratch me, I'm getting a subcutaneous injection with more antigens than my entire history of vaccination over my lifetime in that scratch.
Cool. I think it's funny that Mark asks us if we take questions.
Yes.
Not a listener, I suppose.
We definitely take questions.
Alan, can you take the next one?
Sure. Mario writes, Dear TWiV team, in episode 879, you discuss a Nature paper on altered TMPRSS2 utilization by the Omicron variant of SARS-CoV-2. Given the apparent importance of the presence of a furin cleavage site, which enables TMPRSS2 cleavage of spike in the pandemic spread of the coronavirus, it is surprising that this biological characteristic is now lost in the Omicron variant. Possible reasons for apparent selection of this change in Omicron are not given in the Nature paper and were not discussed in the TWiV episode. My blind virologist guess is that ablation or reduction of cell surface fusion in Omicron infection is a consequence of loss of the furin cleavage. As a consequence of loss of the furin cleavage site, may further contribute to immune escape of the variant. While entry of enveloped viruses by fusion at the cell surface will result in the presence of viral membrane antigens on the surface of infected cells, this is not the case when viruses enter via the endosomal lysosomal pathway. Notably, in the former case, infected cells could be targets for antibody-dependent cytotoxicity, whereby virus propagation could be reduced in a host that has humoral immunity to the virus. With best regards, Mario. That's a reasonable theory, right?
For almost 2 years, it wasn't a problem, right? The virus did quite well, spreading through many millions of people without worrying that there were some antigens left behind when it fused at the surface.
Yeah,
so,
I would worry more about the antigens on the surface at release.
Okay,
the ones that are put up there during reproduction, right?
Yeah, I think these changes are related to evading pre-existing immunity in the population.
I don't think it's likely that that's an issue. I don't know the reason for the loss of the site. It may be that it doesn't matter too much in people, or maybe something else is compensating. I just don't know. I think
I think it's most likely a side effect of
it could be
of this mutation, which maybe helps evade antibody responses a little bit, and that gives it an edge. And coincidentally, this knocks out this other function.
We'll see, perhaps.
Yeah.
Brenda sends us a link to an article in the conversation. China is developing its own mRNA vaccine, COVID mRNA vaccine, and it's showing early promise. Okay, so this is funny because the president of China, if you remember, was highly critical of mRNA vaccines, saying they weren't safe. China, you can find a lot of articles. Here's one in the Washington Post. China turbochargers bid to discredit Western vaccines, spreading virus conspiracy theories. Okay. And now they realize that their vaccines aren't so good. So they're going to join the fray. Okay, China.
You can probably find those archive news stories easily if you're outside of China.
Yeah, I found them very
yes.
easily.
I'm not sure that you would be able to find them quite so easily from the other side of the Great Firewall, but...
Yeah, I agree. So let me take a real email from Flavia. My husband and I have thoroughly enjoyed, learned from, and appreciated many TWiV episodes, even when we struggled to understand terminology weeds used in TWiV discussion. We never use weeds on TWiV, weed, right?
I haven't used weed since college.
Yeah. I just saw on YouTube the following.
Different thing.
Yeah, that's what I meant. That's what I meant. We don't use weed. What did Bill Clinton said? I didn't inhale.
Didn't inhale, right.
Yeah.
I just saw on YouTube the following report, and I'm wondering if, hoping that you all might discuss this report in Nature on a future TWiV, a vaccine pathway for herpes virus with Gregory Smith. He recently published findings in Nature that bring the possibility of a vaccine a step closer. So the paper in question is the motor that gets incorporated into the virus. Remember, we did that on TWiV?
Yeah.
So, Flavia, we did it already. It's TWiV 883. It had a good title, Grand Theft Kinesin.
Yeah.
That was, I think, your favorite title of the year, Alan, right?
I think that might have been, yeah.
And I don't know what the vaccine angle is. You're not going to immunize people against the kinesin because then it's going to mess us up because we need the kinesin. So I don't know what the vaccine angle is. He's been investigating a path to long-needed vaccine development. He recently published these findings that bring the possibility of a vaccine a step closer. I don't know what thoughts you guys might have that this motor brings a vaccine a step closer.
Don't know.
I don't
get it.
Greg, if you're listening. On another note, we recently attended a Café Scientifique presentation of viruses given by Dr. David Beckham of the University of Colorado School of Medicine. After his presentation, we asked him if he happened to be a TWiVer, to which he responded yes, and in fact had lunch with you at the last ASV meeting in Minnesota. We felt so in knowing about TWiV. P.S. All our Amazon purchases are made through Amazon Smile. Thank you for all your fascinating and important microbe TV science facts. Flavia is in Denver, Colorado, or Flavia. I don't know.
Or Flavia?
Flavia or flavivirus, right?
Yeah, I thought about flaviviruses.
And Brian, why don't you take the last one?
Okay. Nikolai writes, hello, TWiVers. I have been a fan since TWiV 720, ever since I bought my first car. I'm a first-year student at the University of Cambridge, studying biological natural sciences with plans of specializing in biochemistry, but this podcast has definitely sparked my interest in virology, which has been helpful in my essays. This month, TWiV has been a major grounding point for me as I've been catching up on older episodes while driving supplies to Ukraine. As a Ukrainian, I felt like I've had to do anything I can to help, and though I've put my studies on pause, I wanted to keep learning even in this turbulent time. So thank you, TWiV, for unknowingly being there for me. Now I have joined the Ukrainian resistance in a volunteer battalion as a medic. Our unit specifically is working on building and operating drones, which will be used to deliver humanitarian aid to places where humanitarian corridors have not been established yet, such as donor blood and vital medication, among other cargo. As we are a volunteer unit, we do not get any funding from the government and have to rely on our own finances, which is why I am currently doing a fundraiser, information about which is on my Facebook and Instagram pages, which you can find by searching my name. Thank you for all of the great work you've been doing for the scientific community. All the best, and since Nikolai mentions his name, Nikolai Nizolov. P.S. I have downloaded some episodes to cheer me up for when I'm closer to the front lines. Well, thank you for doing that, Nikolai. I think that that's really important work, and I guess I'm glad that we're helping you out, but I think that you're doing the really important stuff here.
Can we link to Nikolai's Facebook or Instagram there?
Yeah, I'll look it up and put it in for sure. Facebook and e.g. I have a student in my virology class who's over in Ukraine right now as a medic.
Wow.
And it's her second tour. She was there a couple of weeks ago, and her car got blown up. It ran over a mine. So she came back, and she comes to class all cheery and telling me about this. And now she went back, and each time she has to delay her quizzes and exams. And I said, no problem. Don't worry about it. Just please be safe.
Just come back. Yeah.
Yeah.
Just come back. She just emailed me today. I can't take the exam. The Russians are blocking our internet or whatever, So I said, don't worry. Just take it when you can.
Yeah. And same to you, Nikolai. Come back.
Yes.
Yeah. When you're done there, make it back alive.
Please come back. Please be safe.
Yeah.
So in her case, their car had 6 people in it, made a wrong turn, and it ran over a Ukrainian mine.
Oh.
And the driver had to have his leg amputated, and she said the car was completely destroyed. She doesn't know how everyone survived, but she had trouble hearing in her right ear for a while.
Right.
So you guys are very brave for doing that. So thank you. All right, let's do some picks of the week. Brienne, I know exactly what you got because I was on this site yesterday and saw it.
Yes. So I learned about this last week. In fact, I learned about it immediately after some students in my lab had to submit a poster. Would have been nice if I'd learned about it a day or so earlier. I've picked Biorender before, which has a lot of very useful information or very useful things for making figures and things like that. But this is Biorender's poster builder. So Biorender has made a template that you can use in order to make a poster. It is really nicely customizable in terms of sizes of columns and boxes and colors and just all sorts of things. And I am pretty sure that many of my future posters post last week are going to be made using this poster builder. And it's free on their website.
So I got a Microbe TV subscription to Biorender because I don't like having their name on every piece of art that you export,
Right.
And plus you can get higher resolution when you pay for it. It's really cool. I've done a number of nice figures.
I make a lot of nice figures for classes. I really use Biorender a lot.
I did what did I do? Oh, I did a blog post figure, which was, you remember we did the paper on the virus nucleocapsid. So I wanted to explain the concept of a nucleocapsid. So I took some of their figures, which you can always break up into pieces, right? And did it. It was lovely. It's really lovely. High resolution because I'm a subscriber and so forth. So it's pretty neat. I like it. Although I noticed, though, I was looking and they have a Epstein-Barr virus reproduction scheme where the virus goes in, you have an acute infection, you have latent infection of B cells and so forth. And it looks very much like the figure from Principles of Virology. Yeah. You got it. It's changed enough so that it's different. It's fine with me. I don't care.
They were inspired.
Yes. Inspired by.
No problem. Alan, what do you have for us?
I have, so this is a pick for the cats of TWiV, one of whom is sitting next to the desk, resolutely out of the frame. And any other cats of listeners out there. Ours have found these absolutely mesmerizing. These are Hex Bug Cat Toys. So this thing, it's a little, the one that our cats have, actually they have like 4 of them scattered around the house now under various furniture. It's about the size of the head of a toothbrush, and it's got little bristles coming down, and there's a vibrating motor in it. And so you and then it has a string with a little fluffy tail on it. And you turn on the switch, and as it vibrates, it crawls around on the floor, usually in a circle. Sometimes it'll bump into something and navigate along the wall. And this absolutely drives cats nuts.
I can totally imagine, and I will need to get one of these.
Go ahead and get one of these. It is very fun to watch. Oliver has actually, he doesn't care if they're running. So even when it's off, he likes to fetch them. He's become obsessed with these particular things for carrying around and dropping at your feet. But Suki likes it when it's animated.
It's cute. That little cat there is very cute.
Yes.
Adorable. Neat.
So enjoy.
My pick is something one of our listeners sent, Les, who is also a moderator for our Wednesday night live streams. He purchased this and sent it. It came to the incubator. We get mail now here at the incubator.
Hey,
it's so cool.
You have
so
a
this
real
is
address.
this is a United States Mint American Innovation coin. It's a $1 reverse proof coin, and it's the state of Pennsylvania. So I'm going to open this up for those of you watching. It's got this cool package that opens up multiple ways, and then the coin is there sealed in. And on the front of the coin is a picture of a microscope and polio virus, and it says polio vaccine 1953. So this is a coin commemorating the polio vaccine, which is why Les sent it to me.
Nice.
And there's a little blurb here I want to read because it's cool. In 1947, the University of Pittsburgh hired Dr. Jonas Salk to develop a polio vaccine. At the time, polio was a devastating disease, disproportionately affecting children and young adults, leaving many of them paralyzed. In 1953, the university announced that Salk's team had discovered a vaccine they believed could prevent the disease. After 2 years of trials, the first of which were conducted in southwestern Pennsylvania, the vaccine was determined to be safe and effective. Its subsequent widespread administration to millions of Americans effectively eliminated polio and helped usher in a new era of virology. The discovery of the polio vaccine changed the landscape of growing up and living in America. Prior to the vaccine, the nation was gripped with fear about the ravages of polio. Its discovery lifted spirits and helped return a sense of community and leisure to American families. I thought that was an interesting word, leisure.
Leisure, yes. Well, you can go swimming again.
Yeah, you can enjoy your swimming pools.
That kind of leisure. I mean, I just don't think people have much leisure anymore. It was an achievement hailed by the American Medical Association as one of the greatest events in the history of medicine. I think that's very cool. So
it is.
It's a coin, it's issued for the state because of this, and I like it. Thank you, Les.
And they're doing a series of these for all the states.
Yeah, they have different things for each state. So
New Jersey has a light bulb.
New Jersey has a light bulb. Yes, Thomas Edison. Right. I'm going to put this right here for now in front of the page. We'll find a good place for it. But thank you, Les, for sending that. And that's pretty cool. I'll put the link so if you want, you can go buy it or you can buy others. It's neat. Very cool. All right, we have 2 listener picks. We have Sue. Hi, Twivers. Love your podcast. I just finished episode 869 and heard Rich's pick and the interest in the Illusion Art Museum in Prague. If you don't know about it, there's a similar and rather astonishing museum in Baltimore called the American Visionary Arts Museum. This Tesla piece would fit right in. I've seen things like this in pieces even more astounding. One of the artists we discovered there is Dalton Getty, who sculpts, carves intricate objects from pencil lead without magnification. Their biggest fundraiser is the Kinetic Sculpture Race, slamming science into art in the best, most fun ways. I can't explain it at all, so she gives a link to that. They explain themselves. AVAM specializes in original thematic exhibitions that seamlessly combine art, science, philosophy, humor, and especially social justice and betterment. Go to Baltimore, and thanks for keeping me somewhat sane and educated for the last 2 years. Sue from Pittsburgh, where it's a cloudless, sunny, and crisp 33F 0.6 C. I think this was a couple of weeks ago. 869, yeah. Cool. And then Fraser writes.
That's cool. I want to go
It's a very cool museum. Yes. If I ever go to Baltimore, I'll check it out.
I'm sure I'll be down there at some point. Cool.
I'm a PhD student in virology at the University of Leeds, focusing on hepatitis E virus. I found this article on the BBC that was incredibly interesting since TWiV recently was discussing fieldwork in animal viruses. Now, this is a cool article. Vaccine trial for killer elephant virus begins. Oh, this poor elephant. Veterinary team worked around the clock to save this elephant from the dangerous elephant herpes virus in 2019. Yes, so herpes viruses are quite lethal to elephants. They often kill elephants in zoos, and so apparently they're developing a vaccine. The trial is ongoing. Yeah, so that's cool. A number of people work on these elephant herpes viruses. quite interesting. Keep up the great work. Best wishes from a cloudy 8 degrees Celsius Leeds, recovering from a recent storm. Oh, sorry to hear about that. My notes are all screwing up here. What's going on?
I'm not doing any.
Mine kind of, there's no more cursor.
Oh.
My mouse, when you click, it doesn't put a cursor in. It just, all right. Well, anyway, since that's good.
It says you're
because
clicked on the link.
Yeah, I closed the link, but it's fine. Because we're done. That's the end of TWiV 888, and I don't need to read anymore because I know what comes next. You can find the show notes at microbe.tv/TWiV if you want to send a question or a comment or a pick. We'd love to get that from you. TWiV at microbe.tv. If you enjoy what we do, consider supporting us. You can go over to microbe.tv/contribute and make a contribution in a number of ways. You can do that, and your contributions are U.S. federal tax deductible because we are a 501c3 entity, charity, whatever you want to call it. Brienne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brienne.
Thanks. It was great to be here.
Alan Dove's at alandove.com. He's Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.